So welcome to the RNAi, microRNA, and developmental timing session. My name is Anthony Jose, and Julie Claycomb will be moderating this along with me. I'll do the first 4 talks, and then she'll take over. The first speaker for us today is Hannah Seidel from Judith Kimball's lab from University of Wisconsin.
Okay, thank you. I'm happy to have the opportunity to talk to you today. My talk is going to focus on the effects of starvation stress on the cell cycle of germline stem cells in C. elegans, and that's the cell cycle that you're seeing here in this title slide. So my interest in looking at the effect of starvation on these cells arises in part from some observations about how the presence or absence of food affects the size and morphology of the germline tissue in C. elegans. And that change is documented on this slide. On the top, you're seeing an image of the germline in an adult hermaphrodite that has been fed continuously throughout her life. In the middle is an image from an adult animal that was starved beginning from the final larval stage. You can see that in the starved animal, the germline is much smaller and it has a shriveled, shrunken appearance. This change is reversible upon refeeding because if we refeed such a starved animal, the germline can regrow into something that very closely resembles the germline in a continuously fed adult. I've been interested in understanding the cell biological changes that underpin this major plasticity in the germline tissue. And today I will talk about my work addressing the question of how do starvation and refeeding affect germline stem cells. And I'll first talk about effects on stem cell proliferation, and second, effects on stem cell maintenance. Mitotically dividing germ cells in C. elegans reside in the distal region of the gonad, that's here. And for simplicity in this talk, I'm just going to refer to this group of cells as germline stem cells, although in reality it includes stem cells as well as cells that have begun to differentiate but continue to cycle through the mitotic cell cycle. Under fed conditions, these cells have been studied extensively and they're known to cycle continuously through the cell cycle with a timing of about 6 to 10 hours. And the mitotic cell cycle in these cells is dominated by S phase and G2, with G1 being very short. We have a number of tools for assessing the proliferative state of these cells, one of which is to stain gonads with a marker for cells in mitosis. So we're staining for phosphohistone H3, and this shows what that staining would look like in a fed animal. So on average, we see about 5 to 10 M phase cells per gonad arm. And that's because although these cells are cycling continuously in a fed animal, they're cycling asynchronously, so we only see a few cells in M phase at one time. As a first step to understand how starvation affects the cell cycle in these cells, I repeated this staining in starved animals to ask, what do we see in terms of the number of M phase cells? And before I show you the data from starved animals, I first want to show you what comparable data looks like in fed animals. And here on this graph, I'm plotting the number of M phase cells per gonad arm for a large number of animals. So here, each dot on this graph represents a data point from an individual gonad. Most gonads have between 5 and 10 M phase cells, although the whole spread of data is much greater than that. If we perform a time course in fed animals, we see that over time the number of M phase cells stays relatively constant. This makes sense because these cells are cycling continuously, so at any moment in time there's a new cohort of cells moving from G2 into M phase. But if we perform the same time course in starved animals, we see a really different pattern. So that time course is on the right of this graph. Time 0 is the moment of which I've removed food from these animals. And you can see that just half an hour after removing food, we see a major drop in the number of M phase cells, and then M phase cells essentially disappear entirely during starvation. We interpret this data to indicate that under starved conditions, these cells stop dividing. We next wanted to ask, what effect will refeeding have on these cells? Will they re-enter the cell cycle? So we performed an analogous refeeding time course which is shown here. So here, time 0 is a moment at which I'm adding food back to animals that have been starved for 12 hours. And you can see that just 1 or 2 hours after returning food to these animals, we see a big spike in the number of M phase cells, and M phase cells continue to persist thereafter. Whoops. So we interpret this data to indicate that refeeding causes these cells to resume division. So what I've shown you is that starvation blocks cell division in the germline stem cells and causes them to enter a quiescent state. As a first step to understanding the regulation of this quiescence, I wanted to ask what's happening to progression through other stages of the cell cycle. So under starved conditions, are G1 phase cells continuing to initiate S phase and replicate their DNA, our cells continuing to progress through S phase, and our S phase cells continuing to enter G2. So I've repeated the same starvation time course I just told you about, but instead of counting the number of M phase cells, I instead monitored the proportion of cells in G1, S phase, or G2. That data is shown on this graph. So you can see that, and I'm just plotting the summary statistics here for simplicity. So G1 phase cells, which are infrequent to begin with because G1 is short, they disappear very rapidly after starvation. And we interpret this to mean that under starved conditions, cells do continue to initiate S phase. We also see that the proportion of cells in S phase decreases, and at the same time, the proportion of cells in G2 increases. And so we interpret this data to indicate that cells are progressing through S phase during starvation and then pausing in the G2 phase of the cell cycle. I'll say, however, that the rate of decrease in S phase cells occurs more slowly than we'd expect under fed conditions. So although cells are continuing through S phase, they're doing so more slowly than in fed animals. So these effects on the cell cycle are slower progression through S phase and pausing in the G2 phase of the cell cycle with a block to entering mitosis. And I want to pause here to say a few words about this G2 pausing. So typically when we hear the word quiescence, we think about cells pausing in the G1 phase of the cell cycle before DNA replication. And that's because work in mammalian tissue culture in the 80s demonstrated that most mammalian cell types, or many mammalian cell types, will pause in G1 when they become quiescent, and so we think of quiescence as something that happens in G1. But in fact, if you look throughout the literature in a variety of cell types and a variety of species, you find examples of cells pausing in G2 in response to things like changes in nutrient levels or changes in growth factors. And the emerging picture of germ cells in C. elegans is that whenever they pause, they do so in G2 and not in G1. During the second portion of my talk, I'm going to transition to the topic of stem cell maintenance and look at the effects of cell cycle quiescence on that maintenance. Under fed proliferative conditions, it's known that germline stem cells, for their maintenance, require interaction with a somatic niche cell that sits at the tip of the germline. Contact with this niche cell triggers notch signaling in the germline stem cell, and the active output of that signaling pathway promotes the stem cell fate and inhibits differentiation. And I wanted to ask the question, does cell cycle quiescence maintain stem cells in the absence of notch signaling? So is this signaling pathway also required in the quiescent state? This question is important because in a variety of systems, it's been postulated that cell cycle quiescence is intrinsically linked to the stem cell state, but no one has had the tools to ask, if I induce a quiescent state in a stem cell population, will that remove a requirement for the signals that are required under proliferative conditions? And so now that we have this ability to induce a facultative cell cycle quiescence in this cell type, and because we also have a lot of tools for manipulating notch signaling, I'm able to address this question in this population of cells. The tool that I'll use for manipulating notch signaling is a temperature-sensitive mutation in the notch receptor. This mutation causes the protein to be functional at 15 degrees, the permissive temperature, and non-functional at the restrictive temperature of 25 degrees. As a result, Under proliferative conditions, there's a difference in stem cell maintenance at these temperatures. At 15 degrees, these animals maintain their germline stem cells, but if we shift them to 25 degrees, those cells differentiate. And we can assess that differentiation very easily because the first step of differentiation, which in this case is entering the meiotic cell cycle, is that the chromosomes congregate on one side of the nucleus, giving the chromosome configuration a very distinctive crescent shape. So I've repeated this temperature shift experiment under starved conditions where these cells are quiescent to ask whether Notch signaling is also required under quiescent conditions. And I'll show you that data on this slide. Remember that I have two conditions here, fed conditions when the cells are proliferating, starved conditions when the cells are quiescent, and for these two conditions I'm looking before and after the temperature shift. And the way I'm plotting this data is to count the number of rows of germline stem cells, which is simply the number of rows counting from the distal end of the germline. So for fed animals at 15 degrees, we see those animals have around 10 to 13 or so rows of germline stem cells, but when we shift them to 25 degrees, all of those stem cells are lost, and so this population drops to the 0 category. When we repeat this same experiment in starved animals, we see a different outcome. Here we see that even at the restrictive temperature of 25 degrees, those cells are maintained in what looks like a mitotic cycling state. So the comparison here is these two categories of data. To really ask whether these cells have been maintained in a stem cell state, I wanted to ask whether they also retained two other properties. One is the ability to divide mitotically, and there the answer is yes. If I refeed such animals, those cells will re-enter the mitotic cell cycle. And second, I wanted to ask whether these cells are capable of long-term maintenance. And again, the answer is yes, because if I return these animals to 15 degrees and refeed them long-term, which in the worm is 2 to 3 days, the stem cells are maintained. So we conclude that under conditions of active proliferation, although Notch signaling is required for stem cell maintenance and required for preventing differentiation, that starvation can stand in for the requirement for Notch signaling and thus renders Notch signaling dispensable. I also wanted to ask, is this effect of starvation something specific to starvation per se, or is it caused by the cell cycle quiescence induced by starvation? So I repeated these same experiments under two additional conditions that affect the proliferative state of these cells. One is the absence of sperm, and the second is high salt, which is an osmotic stress. Both of these conditions reduce stem cell proliferation, although not as severely as starvation. And indeed, under both conditions, we see that Notch signaling is dispensable for the maintenance of germline stem cells. So we conclude that it is really not starvation per se that's allowing Notch signaling to be dispensable, but it's instead the cell cycle quiescence. So to conclude, I've shown you that there's a dramatic tissue plasticity in the germline tissue of C. elegans mediated by changes in the cell cycle of germline stem cells. These cells become quiescent under starved conditions. That quiescence removes the requirement for Notch signaling, and if we refeed these animals, the germline will regrow, the cells will begin dividing again, and the requirement for Notch signaling resumes, which is data I didn't show you. And so I'll end by thanking those people who've supported me, my PI, Judith Kimball, and a wonderful staff scientist in the lab, Sarah Crittenden, as well as other staff members of the lab, and my funding from the Ellison Medical Foundation. Thank you.
Time for questions.
Yeah, is this on?
Yeah.
I wonder if I know that the allele, did you use a single TS allele for GLP-1? Q224 or something?
Yes, I have repeated it with another TS allele, and I see the same thing. I will say the data that you saw today is the strongest of all the TS alleles that we have.
So I wonder if starvation sort of rescues that allele to some extent.
So that's a good question. So one hypothesis is that starvation is maybe upregulating a chaperone that's allowing the protein to be folded. And so we've looked at a readout for Notch signaling in these animals, looking at transcription of a target gene of Notch using single molecule FISH against an intron, which allows us to see primary transcripts. And we see that transcription is off at that high temperature.
What happens to LAG2 under these conditions? Have you looked?
I have not looked at that. We heard a talk this morning about the effects of downregulating TGF-beta signaling and its effects on LAG2 expression, and that itself is affected by food availability.
So if you were to artificially block cells in G2,
Yep.
right, in the low-notch condition, would you have the same outcome?
Yes, so we have repeated these experiments knocking down cyclin-dependent kinase 1, which will cause such a block. I've also repeated these experiments when the cells are treated with hydroxyurea, which arrests them in S phase. Both of those conditions do not recapitulate this effect. So it's not just an effect of not being able to, say, go through a G1. It really is something about the quiescent state.
I think we're out of time. Let's thank the speaker one more time.